Assalamu alaikum, my name is Malak Alkhatib, and today we'll be going through the nitrogen metabolism chapter, which is also known as the urea cycle. In addition, we'll be going through the amino acid metabolism, which includes the synthesis and the degradation pathways, which are very easy, especially that this chapter or these two chapters are actually, in fact, they're two separate chapters, but for the sake of making it easy, our doctors have combined them into one chapter. Okay, now you'll notice that as we go through this chapter, I'll be using the question or the problem and the solution approach method, which makes it easier to understand each step. Okay, so let's start off by talking about the amino acid utilization. Now we know that amino acids have a lot of roles, diverse roles, such as protein biosynthesis, energy production, and the list goes on. But to start off with our first process that we'll be talking about, I'm gonna present the first problem. So our problem here is that this amino acid cannot be utilized due to this amine group. And what I mean here by utilization is whether synthesis or degradation, specifically degradation. So amino acids can never be degraded or even utilized generally unless this amine group is removed. And the process of removing this amine group is called deamination. So let's start off by talking about the deamination of amino acids. So basically, when we deaminate this amino acid, which means we remove the amine group, we'll be left over with this carbon skeleton. This carbon skeleton is known as an alpha keto acid. So now we can analyze that an alpha keto acid is basically an amino acid minus the amine group. Now, over here, we can utilize the amino acid. طيب, let's talk about how is deamination specifically done. Now, again, deamination is a reversible reaction, meaning that we can go and then come back to the original structure. And deamination is usually the removal of the alpha amine group from the amino acids, not from the R group. Okay, so deamination is basically done over two steps or can be done by either steps depending on the amino acid that we're dealing with. So we have the oxidative deamination. Along with the oxidative deamination, we have the transamination, again depending on which amino acid or structure we're dealing with. Basically, in our chapter, we have three main amino acids that are usually deaminated, which include alanine. And once we remove the amine group, we'll be left with a pyruvate, which is basically the alpha keto acid. Glutamate, once deaminated, will become alpha keto glutarate, and aspartate, once deaminated minus NH, will be left with the oxaloacetate. طيب. Usually, glutamate. So let's start off with glutamate. Now, glutamate undergoes the oxidative deamination. And why is that? That's because it has the required enzyme that is capable of deaminating it, which is known as glutamate dehydrogenase. So glutamate becomes an alpha keto acid, which is an alpha keto glutarate, by the glutamate dehydrogenase. Now, once we deaminate glutamate, the ammonia will be released, and that presents a big problem. And this problem will be solved by the urea cycle, but that's not our topic for now. We'll go back to this problem. So now we're done with glutamate, and we're left off with the alanine and the aspartate. How do they undergo this deamination? The answer is that they undergo this deamination by the transamination process. And the main reason why they undergo transamination and why not the oxidative deamination is basically because they do not have the enzyme required to directly deaminate them. So we said that we do not have this enzyme for these two amino acids, which are alanine and aspartate. Therefore, we go through other alternative pathways, and this pathway we mentioned, it's called the transamination reaction, which utilizes a group of enzymes. They're called transaminase enzymes. So since we're dealing with alanine and aspartate, we have the alanine and aspartate transaminase enzymes. Now, these enzymes are usually presented in the liver. However, you, as a future physician, if you detect these enzymes in the blood, this indicates that we have a liver damage due to the leakage of these enzymes into the blood. Okay, so according to the name, or as the name presents, as transaminases, these enzymes, which are the AST as a shortcut, AST and ALT, which stand for aspartate transaminase and the alanine transaminase, basically what happens is that we will have our enzyme and it will be converted into an alpha keto acid, but not directly. So what will happen is that we have two substrates, which are the amino acids that we are dealing with, and this amino acid we need to deaminate it. Therefore, we call it as the donator amino group, okay? Or it's gonna give its amine group. So it's gonna donate this amine group, and therefore another structure, which will be our alpha keto acid, which does not have the ammonia, will accept this amino group. So how is that? Let's start off with alanine. Alanine, in order to be converted into a pyruvate, which is the alpha keto acid, it needs to have along with it a ammonia acceptor, or the NH2 acceptor, which will be usually our alpha keto glutarate, which we discussed, which is the alpha keto acid of the glutamate. So basically, the reaction will start off by the alanine, which is our target here. We need to convert it into pyruvate, so it will donate its ammonia to, or the amino group, to the alpha keto acid, which is our alpha keto glutarate. So what will be the result is that we're going to have the pyruvate, which is now deaminated version of the alanine, plus an amino acid, which is our glutamate, since we have added the amino group to the alpha keto glutarate to convert it into a glutamate. And this was done by the help of our alanine transaminase enzyme. Now, the other step, which is basically literally the same thing, the same step, aspartate, it will again donate its amino group to the alpha keto glutarate using this enzyme, which is our aspartate transaminase, and this will result in our alpha keto acid, which is our target, plus the glutamate amino acid, since the amino group was added to the alpha keto glutarate. So now we have discussed this major topic, which is the deamination of the glutamate, alanine, and aspartate, which go through two different pathways. Now, if we go back to our problem that we have discussed initially, which is the deamination of glutamate. Now, we mentioned that glutamate, once it's deaminated by the glutamate dehydrogenase, we get our target, which is the alpha keto glutarate plus ammonia. Now that presents a very big problem, because if you have noticed, when we deaminate alanine, we do not present with any free ammonia in the blood, and that's because the ammonia was incorporated into alpha keto acid to give us this amino acid. Now again, why is that a problem? Why don't you want ammonia in our blood, free ammonia? That's because ammonia is very toxic in the blood, okay? And we know that ammonia can be produced in many different tissues, whether the brain, the muscle, and that presents also a very big problem. So in order to resolve this problem, we need to have a solution, which is basically converting this ammonia into urea, which is also toxic but presents with less toxicity than ammonia. Okay, and this step, which is the conversion of ammonia into urea, is known as the urea cycle or the urea synthesis. So how is that done? Now, before we can proceed directly into our urea cycle, we need to transport this ammonia from the blood to the liver, because this free ammonia that was produced was in the blood, which is very toxic, and we need to convert it to urea in the liver using the urea cycle. So in order to do that, we need to have transporters, because again, ammonia is very toxic, and there are two main transporters which are very efficient that we use. These include asparagine and glutamine, but majorly we use glutamine. Okay, so you may ask, how is that done? Okay, so in order to transport this very toxic free ammonia in our blood to the liver, in order for the urea cycle to occur, because the urea cycle takes place in the liver and this ammonia needs to be reached or to reach the liver, so we have transporters. So let's start off with glutamine. So we know that this is the normal structure of the glutamate, which means that it has a place to accept this free ammonia to incorporate it. And once we incorporate the amine group, the free amine group, which was produced by the deamination of the glutamine, now we're gonna have our transporter ready to go from the blood to the liver. Now this process is done by the help of a certain enzyme, which is the glutamine synthetase, from its name, because we're synthesizing the glutamine, and therefore it's called the glutamine synthetase. Now, if you noticed in this process, unlike the deamination, in the process of transportation, this amine group is incorporated in the R group of the amino acid and not the alpha carbon or the main skeleton of the amino or the amino acid generally. Okay, so as we mentioned, now this transporter is ready to go to the liver. Once it reaches the liver and we have glutamine successfully transported this ammonia to the liver, this reaction is reversible, however, not by the same enzyme. So we can go back from glutamine to glutamate in order to release this ammonia, which needs to go to the urea cycle in the liver. We need to have another enzyme, which is the glutaminase. As its name suggests, it's A's, so we are removing a certain structure, which is the ammonia. And now we have free ammonia in the liver, where we can have our urea cycle take place to convert this ammonia, which is very toxic, into a less toxic compound, which is the urea. Now, as I mentioned earlier, we also have another amino acid that acts as a transporter, but this amino acid is not very preferable and we don't usually use it. And that's because it does its process with the help of glutamine, so it's not independent. So how is that? So basically, again, aspartate is free over here, so it can incorporate this free ammonia, and we do this by the help of another enzyme, which is the asparagine synthase. And now we have the asparagine ready with the ammonia group or the amino group in its R group again, and not in the alpha carbon, the main skeleton. But the problem that presents over here is that the aspartate, when it wants to incorporate this ammonia in order to transport it and to become the asparagine, it uses the glutamine as a help. So it acts as also a donator of the amine group. So basically, this reaction, how it takes place is that the aspartate becomes an asparagine by the help of this enzyme, which is the asparagine synthase, in addition to incorporating this glutamine to incorporate and add this amine group. It's a mouthful, but it's very simple. So it's not a free amine group, not a free one. However, it's one that is found in the glutamine. It adds it to the aspartate, and now we have the asparagine, which acts as a transporter from the blood to the liver, where the urea cycle takes place, and then it's a reversible reaction. It can release its ammonia just like glutamine did in the top over here when it was released as glutamate and ammonia. So let's do a quick recap. We have discussed how deamination occurs, whether it is oxidative deamination or the transamination, depending on which amino acid we're dealing with. And then secondly, we started talking about how the oxidative deamination of the glutamine presents with a problem, which is the free ammonia in the blood, which needs to be converted into urea. And in order to convert it into urea, it needs to be transported from the blood to the liver to be able to go through the urea cycle, since it's very toxic. Okay, so that's basically, again, the oxidative deamination of glutamate to alpha ketoglutarate plus this free ammonia, which presents a problem, makes us start off with the urea cycle. Okay, and this is basically the deamination process. And then over here we mentioned that the preferred transporter is the glutamate to become a glutamine, and then goes to the liver to release the ammonia there. And similarly for the aspartate, which becomes an asparagine. Now, a very important point before we start talking about the urea cycle, I don't like to put things or like to complicate stuff and put them all together. Therefore, I've put it in a separate place. So we know that as transaminations, or like transamination reactions, or specifically the transaminases enzyme, which is the aspartate transaminase and the alanine transaminase, they require a coenzyme, which is the pyridoxal phosphate, also known as the vitamin B6 derivative. Very important to know, and not only in this pathway, but even other metabolic pathways, you need to know the certain coenzymes and the required prosthetic groups for each enzyme as prescribed by your doctor. Okay, so let's start off by talking about the urea cycle, which is our major topic for today also. So we need to navigate properly throughout each step in order to understand this cycle, because it may seem a bit overwhelming as like the diagrams suggest, but if you navigate through easily, it will be very simple to understand. So let's start off by talking about the main aspects of this cycle. So number one, we need to know the location. Where does it occur? Basically, it occurs in the liver. That's our main location. However, it occurs in two distinct areas in the liver, which are the mitochondria matrix plus the cytoplasm. Now, you need to know that the first two reactions occur in the matrix, and then the remaining rest, which are the three reactions, occur in the cytoplasm. This makes our overall number of steps in this cycle 5. So we have 5 major steps in the urea cycle. 2 in the matrix and 3 in the cytoplasm. Another important aspect, the regulatory step, it is the very first reaction number 1. And we need to know that the number of ATPs consumed are overall 4. 2 ATPs in the first reaction and another 2 ATPs in the third reaction. Another aspect, the 3 main substrates that we start off in the urea cycle are ammonia, aspartate, and carbon dioxide. Now, aspartate may seem confusing because if we look at the cycle, we do not see aspartate, but you will know what is exactly meant by using aspartate in this cycle as we reach this part. Okay, so the main aim of this cycle is to convert the ammonia into urea, as we mentioned earlier. A very important step over here to understand is the urea structure. Now, the urea structure consists of the carbonyl skeleton plus the 2 nitrogen. So that's our carbonyl, and then on either side we have the nitrogen groups, which forms our urea. Now, the sources of these 2 nitrogen groups, when we want to form the urea from the ammonia, are from they're different resources or different sources. So the very first nitrogen that is incorporated to form the urea is from the very first reaction number 1, which starts off with the free ammonia. The second nitrogen source comes from the aspartate, which is incorporated in the cycle, as we will see now. And then once we form this structure, we're done with the urea cycle. So another aim that we can analyze is that the other aim is forming the structure, the NCN. Okay. So before we start talking about the urea cycle in details, let me just shed the light on certain conditions that you may need to know, which are clinical correlations. Basically, number 1, we have hyperammonia. In short, it is elevated levels of ammonia in the blood, which is sometimes toxic. Okay, the other condition is known as the kwashiorkor disease / malnutrition. Now, this condition is known to be a negative nitrogen condition, meaning that it's very rare, number 1, very rare, and it also means that the amount of nitrogen intake is much less than what is being secreted or excreted from the body. So, in short, again, it's like an imbalance between the nitrogen losses from the dietary sources, from what is being taken and whatever is lost. So our loss here of nitrogen is much more than what is gained through diet, which suggests that we have malnutrition, less nitrogen in our diet or food, and therefore we also secrete or excrete nitrogen, which gives us this negative balance of nitrogen. Okay, now let's start off with the urea cycle. Bismillah. So first of all, we know that this urea cycle occurs in the liver. Let's start off with the first part, which is the mitochondria matrix. Again, as we mentioned earlier, we have two main reactions that occur in the matrix, which are number one. Let's start off with reaction number one, which are basically the free ammonia, which is our problem, plus the bicarbonate, which is like an analog for carbon dioxide. So bicarbonate plus ammonia will be converted into this structure, the carbamoyl phosphate, and this indicates that the type of reaction number one is condensation, since we started off with two substrates and then we ended off with one. Now, some points to point out in this reaction, which is very important, is that number one, ATP in reaction one is consumed. We have two ATPs consumed, plus it's a regulatory step. Another thing is that the very first nitrogen is synthesized, since we said that this is the normal structure of urea, which is NCN, two nitrogen and the carbon in the middle. So the very first nitrogen is synthesized via reaction number one, and the source is the free ammonia. So that's it for reaction number one, and then we go throughout this whole cycle in order to generate the other nitrogen. Okay, now for reaction number two, we start off from carbamoyl phosphate, which will be converted into the citrulline, and we know that citrulline is a non-standard amino acid. It's not among the 20 amino acids that we have studied. This occurs via this enzyme, which is the ornithine transcarbamylase. Plus, throughout this reaction, something very important happens is that the ornithine gets incorporated into this reaction, which is another non-standard amino acid. So in reaction number two, a non-standard amino acid is incorporated, which is ornithine, plus a non-standard amino acid is produced. So now that is for reaction number two. Reaction number one, reaction number two, we're done. Now for citrulline, it gets transported. It is the substrate or the compound that gets transported from the cytoplasm to the matrix. And then we will start off with our reaction number three, which is the conversion of citrulline to argininosuccinate. From citrulline to argininosuccinate by this enzyme, which is the argininosuccinate synthase, because we have synthesized this compound. Now, something important to note here in reaction number three, two things. Number one, we have incorporated aspartate, which is formed by fumarate, and we will talk about how does that occur. So aspartate gets incorporated in reaction number three, plus two ATPs get consumed, meaning now that we have achieved the 4 ATPs that are supposed to be consumed throughout the urea cycle. Okay, now for argininosuccinate, by now we have the other nitrogen incorporated, which was from aspartate. So now we can notice that the second source of the nitrogen, which is a part of the urea cycle, is formed by the aspartate when it gets incorporated. So now we have our urea cycle. Now you may wonder, so now if we have our urea cycle, our urea compound, why don't we stop till here? Now it's because this the urea substrate is not fully ready. We have other structures that need to be removed by two other reactions, which are reaction number 4 and reaction number 5, and then we will have our urea free. So in reaction number 4, argininosuccinate will be converted into arginine by the argininosuccinate lyase. Now, as you can see or notice that we have released a fumarate in this reaction, and that is one of the compounds that needs to be removed. So if you notice again, ornithine here was incorporated, which are the pink ones. Look at the pink words. So ornithine was here incorporated, aspartate was incorporated, and fumarate over here in reaction number 4 was released. And when did we have our skeleton ready? It was in argininosuccinate, but still we need to continue to remove these two structures from the urea structure. Now we have our arginine, so we said we have arginine. Now here is our final step, the conversion of arginine to ornithine plus urea cycle over here gets released, and that is our target. Now this ornithine that was formed, it re-enters from the cytoplasm into the matrix in order to be incorporated in reaction number 2, which we discussed previously. So now we can analyze that we have two non-standard amino acids that are being antiported, which is ornithine. We said ornithine enters into the matrix and citrulline exits the matrix into the cytoplasm. So something here important to note. Another thing to note is that the urea that we talked about that was formed from the urea cycle, we said that each nitrogen comes from a different source. The very first ammonia came from reaction number 1, from the free ammonia, okay? And the second nitrogen group was formed by the aspartate, which was incorporated in reaction number 3. Now, as I mentioned earlier, if you remember, I said that the aspartate over here is formed from the fumarate that is released from here. So how is that? So basically, once fumarate is released, in order to get this urea cycle to proceed, it needs to form the aspartate. How is that done? If you notice over here, they're exactly the same compounds that are used in the TCA cycle and even the same enzymes, but one thing differs is that the isoform of this enzyme is different. So basically over here we have the cytosolic isoform because these reactions occur in the cytoplasm. However, in the TCA cycle, we had the mitochondria isoform of these enzymes. So fumarates, these reactions, they occur not you cannot see them in the urea cycle, but they do occur in this process. So we have the fumarate, which was released, comes, and then it gets converted into malate using the fumarase enzyme, which is the cytosolic isoform, and then malate will be converted into oxaloacetate by the malate dehydrogenase, just like the enzyme in the TCA cycle, but with a different isoform, which is the cytosolic isoform. Finally, when we have oxaloacetate, it gets converted into aspartate. Now this step does not occur just like in the TCA, it's just a bit different. So oxaloacetate gets converted into aspartate by another reaction, which is the addition of the NH3 or the amino group. Okay, so it's the opposite of deamination. And if you notice that this oxaloacetate is the alpha keto acid that we discussed, which gets converted into an amino acid aspartate. Then once we have aspartate, it gets incorporated into the cycle, then goes on, then fumarate goes out, and then again this cycle occurs. Okay, so by now we're reaching to the end of the chapter, starting off with the amino acid degradation. Now we have initially talked about the first step of the amino degradation, which is the deamination. And because we mentioned that in order to utilize any sort of amino acid, whether synthesis or especially degradation, the very first step is the deamination. So once deamination occurs, which is the process of removing the amine group or the amino group from the amino acid to be left off with this with the alpha keto acid, which is considered as a carbon skeleton, we need to degrade it further. So what do we do is that it gets converted into a certain intermediate, one of the 7 intermediates that I'm going to be listing. So these are intermediates of further glycolysis or the TCA cycle. Now these intermediates include the pyruvate, the oxaloacetate, the alpha-ketoglutarate, the succinyl-CoA, the fumarate, the acetyl-CoA, and then finally the acetoacetyl-CoA. Now if you notice, this intermediate is none of those intermediates, none of both pathways, however it is there. So now, according to the fate of the amino acid, how does it end up, or where does it end up, or to what intermediate does it end up? According to its fate, basically, it can be classified as whether ketogenic or glucogenic. So what do you mean by ketogenic? Basically, any amino acid that gets degraded into whether acetyl-CoA or to the acetoacetyl-CoA, we call it as ketogenic amino acids, and we have only two, which are the lysine and the leucine. The rest are glucogenic amino acids. However, do not think that because we have two ketogenic, meaning the rest of the, let's say 18 amino acids that we have are glucogenic. No, because we also have other amino acids that can be both ketogenic and glucogenic, which we will discuss. But for now, as I said, we have the ketogenic and the glucogenic. Now, we classify certain amino acids as glucogenic if they end up by the five first intermediates, whether pyruvate, oxaloacetate, succinyl-CoA, alpha-ketoglutarate, or fumarate, it's glucogenic. If they end up as whether acetyl-CoA or acetoacetyl-CoA, we call them as ketogenic. Now we're going to discuss by the end what exactly are the glucogenic amino acids, but for now know that the ketogenic amino acids are only two, lysine and leucine. Let's talk about a very simple part of this chapter, which is the amino acid synthesis. It's much more easier than what we think. So basically, in order to synthesize any amino acid of the 20 standard amino acids that we have, there are 6 families that we start off with. So we have the aspartate family, the glutamate, the histidine, the serine, the aromatic family, and the pyruvate family. Each give rise to a certain amino acid or even a keto acid, plus some of them come from a certain precursor, as we will see. Okay, let's start off with the serine family. Now, for the serine family, as you can see, this is our serine amino acid, which can give cysteine and glycine. However, the precursor of serine over here that you need to know is glycerate 3-phosphate, and that's it. Number 2, for the pyruvate family, we have the pyruvate, which can give rise to 4 non-polar amino acids. So that's this way it will make it easier for you to understand and memorize those 4 amino acids. Just know that they are those 4: the isoleucine, the alanine, the valine, and the leucine. And we're done with pyruvate. Now for the histidine family, very simple. What gives rise to the histidine is a certain precursor, which is the ribose 5-phosphate, which is seen in the PPP pathway. Okay. Now for the glutamate family. Now the glutamate, just know that it gives rise to 3 compounds, which are the proline, arginine, and the glutamine. Plus the precursor of glutamate, or what gives rise to glutamate, is the alpha-ketoglutarate, which we know upon not deamination but by the reverse reaction. Now we have the aspartate family. You need to know that the aspartate gives rise to these 4, which are the lysine, methionine, threonine, and the asparagine, or the asparagine. In addition, you need to know that this aspartate comes from a certain precursor, which is the oxaloacetate, which we already have discussed, along with the glutamate, which comes from the alpha-ketoglutarate. Now, according to your doctor, just make sure which of these families you need to memorize, or if you need to memorize all of these amino acids. But generally, they're very easy to memorize. So to be on the safe side, know them. Now, finally, for the aromatic family, we have it's kind of like a different pathway, but still the same concept. So the precursor of this family is the phosphoenol pyruvate, seen in the glycolysis pathway or the glycolytic pathway, which gives rise to something called the chlorismate. Okay, now we just know that this compound will give rise to the phenylalanine, the tyrosine, and the tryptophan, which are those 3 amino acids. And notice all of them are aromatic because they come from the aromatic family. Okay, so now we're done from the urea cycle, as we mentioned earlier, then the amino acid degradation and the amino acid synthesis, which is pretty easy and direct. Now let's just cover something very simple, is the utilization of these intermediates. So you need to know that when a compound gets degraded into whether acetyl-CoA or the acetoacetyl-CoA, it is used for one thing. It cannot be used for glucose synthesis. However, it is used for the formation of ketone bodies. Another thing is that if it's converted into the 5' intermediates, it can go through the glycolytic pathway eventually, or even to synthesize glucose generally. Now, again, just a quick recap. We said that the amino acid catabolism or the degradation begins with the deamination, which is the removal of the amino group, which will be used in the urea synthesis. And then further, the carbon skeleton that we are left with will be degraded into one of the 7 metabolic products or intermediates that we talked about. And then according to the fate, according to which intermediate it is converted into, we can classify the amino acid as ketogenic or glucogenic. Now for the parts. We mentioned that the alpha ketogenic amino acids are only 2, which are the lysine and the leucine. We cannot say that the rest of the 18 amino acids are glucogenic, because 13 of them are strictly glucogenic, which means you can say that they are glucogenic. However, 5 of them are mixed. It could be whether ketogenic or glucogenic. So, in order to make the memorization process easier, just remember that the ketogenic are 2, leucine and the lysine, and the mixed, or which could be whether glucogenic or ketogenic, are 5. Isoleucine, phenylalanine, threonine, tryptophan, and tyrosine. Then the rest of the amino acids are glucogenic. Do not memorize them, memorize these, and then if the question in the exam comes that you're not familiar with the amino acid that you did not memorize, this means that it is the glucogenic. Okay, so that's what you need to memorize. Now, another thing is that we need to know which amino acids are essential and which are non-essential. Now, when we talk about essential amino acid, an essential amino acid is basically an amino acid that cannot be synthesized by the body. However, it could be taken by dietary products. However, the non-essential amino acids are the opposite. Those are the amino acids that can be synthesized by certain biosynthetic pathways in our body. Now, to make it easier for you, just memorize the essential amino acids, which are 9 amino acids. You can use this mnemonic, which is the melting point very low. So you can say kind of that this is the melting point and this is very low. So the TT, which stands for the threonine, the tryptophan, then the HI, the histidine, and the isoleucine, and then the MP, VLL, and so on and so forth. But just make sure that the table that you are provided with in your PowerPoint corresponds to this one, because sometimes certain changes can be done in the classification process. Okay, so for now we're done. Let's do a very quick recap. So we started off by mentioning why do we need to deaminate any amino acid in order to be utilized, and we talked about the process of the deamination, whether it is by the oxidative deamination or the transamination reaction, depending on which amino acid we're actually using. Plus, then we talked about the problem which presents by the oxidative deamination of the glutamate due to the ammonia or the excess ammonia. Therefore, we then went into the urea cycle when we talked about the transportation process, reaching to the urea cycle, all of the rules and stuff we need to know. Then we talked about the amino acid degradation, the steps. Then we talked about the amino acid synthesis, and we ended up with the classifications, whether ketogenic or glucogenic. Plus, we also talked about whether this amino acid is essential or non-essential. And by this, we are done. Thank you. والله يوفقكم كلكم يا رب.